I was asked to give an overview of gene drive so that people maybe can have a better grasp of what we're talking about. So I had some slides I think I've given at UVRI, and so I'm going to use those so that at the end of the day we can confidently say what gene drive is at least, and we can see how it can really help us in the fight against malaria vectors. So those two things, and hopefully we can split the hairs between gene drive and just normal GM. So if those three things are achieved, then we'll have advanced. So of course, okay, myself, I'm from the Uganda Virus Research Institute. I'm a principal investigator there, and I run target malaria activities in the country. And my background is genetics of disease vectors and pathogens. So of course the reason we are thinking about gene drive is because of the malaria, and we know we can't say enough about it. It's still a problem for us. The good things, we also know a lot about malaria. For example, we know the cycles, we know how the whole pathway of transmission runs, and so we know there are many targets which we can control if we wanted to, which we can target if we want to control the disease. So for disease vectors, of course we target the mosquito that transmits it, but then there are other efforts that are targeted elsewhere. For example, against the parasite directly, and also medications in the people. But whatever we do, whether targeting the mosquito or targeting the other stages of the life cycle, we are encountering challenges. Yeah, we are encountering challenges with it because the tools that we're using by themselves seem not able to eliminate the disease. Remember, we have some elimination targets that we signed up to, at least most countries are aspiring to. These have been in place since 2016, and we had goals, eradicating or lowering the mortalities and morbidities by a certain percentages, and we hope by 2030 we would have done so by 90%, based on the 2015 levels. But if you look at that trajectory, at the moment, we are really lagging behind, way behind, and we're not, anywhere near, attaining that. And we know that the control gains that we've been making are beginning to slow down, and this has been apparent since 2015, 2016. So that means that if we are to put our targets back, our elimination targets back on track, we need to do something differently. We need new methods that can complement what's already existing. So that's the idea of looking for additional things that we could use in the fight against malaria. So here is just a projection by modelers when they look at what could happen if we just scaled up what we have. That was 2016, increased it in many folds. They modeled different scenarios, but I'm just pulling out one of the more optimistic one, whereby they increase things, say, fourfold. So increase the funding fourfold, the coverage and that kind of thing, and at the same time keep the research funds available to get new tools. So if that was done, you could still see that by 2030, you'll still not have eliminated the disease. Yeah, some areas could have benefited, and you could have achieved local eradication, but not elimination. There'll still be places with lots of malaria, and most of that is in Africa. So then that means that clearly we need to do something beyond just the current tools that we are having. And of course, then that's where gene drive comes in as one of the options that could be harnessed for the control of malaria vectors. So the idea that chemical control, which is the mainstay for vector-borne diseases now, you spray or you use nets if it's malaria, has been around for quite a long time, but at the same time they've always been thought about complementing it with other measures, including even genetic modification. But it couldn't be done for a long time because of various issues. Mostly it was the technology, so we didn't know enough about, if we were to use a genetic modification approach, the location of the genes that we are going to harness or to use, where were they in the genome? So that's a scientific thing that needed to be worked out. It had to wait until the whole genome sequence was done in the 2000s, and then it was annotated, characterized, and then we got to know the locations of genes, because you have to know the genes you're going to use and where it is so that you can target it. So that was one technological limitation. The other limitation, which is related to that, is once you modify, then you need to find a way of spreading your modification quickly through, the wild mosquito population, if you're going to use that modification as a tool. So then the question was, how do you do it relatively quickly in a short period of time so that you start, harnessing the gains? So that second part was a challenge, and for a long time, we had no clue. So it still remained theoretical that you could use it, but it wasn't clear how it could be made into an effective tool that could spread quickly. So then that's where GeneDrive came in. So GeneDrive came to fix that other second part, and I'll show you how. So maybe just to recap, so if you're going to modify, you are thinking about the stepwise how you do that, but the two things that you need to have clear before you start is, one, it's quite specific, so you need to decide what changes you're going to effect in the mosquitoes. And broadly, these are really the changes, or what you call factors that facilitate the mosquito in transmitting the malaria. So there are many that you could target. So if you zero in on any of those, you have a candidate, you can proceed. Then number two, once that's done, you are still not yet through, because you need to spread that quickly through the population. Then you have to figure out how to spread those modified changes quickly through the natural mosquito populations. So those are the two things that you have. So then what can be targeted? In practice, many things, so you could target the behavior. So for example, you could say, you could modify and change the biting preference, make it maybe not bite people, bite other things, or you could attack vector susceptibility. You make the mosquito immune to picking up the parasite, or the longevity, you reduce the lifespan, or the numbers, many things you could target. But right now in the field, the most advanced measures are those that are targeting the susceptibility. This is the anti-parasite strategy, and then those that are targeting the numbers, whereby we're trying to reduce the numbers that spread the mosquitoes. So these are the two leading efforts that you're going to hear about. It doesn't mean people can't develop those others, but in terms of progress, how far the field has gone, the teams that are working in this space are either working on this or they're working on the other one, okay? So again, this is a recap of the other one, just in cartoon. So what could useful modifications be? So I've zeroed down now on the two that are most advanced, where it's practical, where we can dream and see what could happen. So you could modify to suppress the population, that is to reduce the numbers that bite. And for that, you could target, for example, female infertility. You could modify to induce it. That can be one approach. Or you could modify and skew the male sex ratios among the progeny through the generations. And if you did that, you can imagine that you'll be having more males at the expense of females. It's the females that really transmit, and it's also the females that determine the number of the mosquito population in the next generation. So if you kept reducing that, a time comes when there are so few females that you can break the transmission. So this is the suppression approach, two flavors of it through genetic modification. And then the other one, anti-parasite, you could of course modify so that you make the malaria not be able to pick up the parasite. So you make the mosquito not what the technology, in terms of, they call it refractoriness. So you introduce refractoriness in the mosquito. So it can pick up the parasite, but the parasite can't establish itself or complete the life cycle. So if that happens, then you can't the mosquito then can't transmit. So if you had those kind of mosquitoes that they were the ones that are flying around, then you can theoretically curb malaria transmission, okay? So these two strategies, genetic modification. So now we've said that you can modify in principle, and you can do it in the lab, and you can create one that's able to, say, maybe be infertile, or one that's resistant to malaria parasite. And even if you did that and you had it in your lab, there's no reason why that trait, if you introduce it in nature, is going to spread, because that's not how biology works. So you still have a problem. So how do you quickly spread that trait relatively quickly through the wild mosquito population, so that they too, if it was anti-parasite, trait that you had introduced, they too have it, because that's the only way. Because you're modified when it's just the few in the lab, but out there are those ones which are transmitting. So you need to spread this to them. So how do you do it? Relatively quickly. So that's where gene drives come in. So I want you to distinguish that gene drive, yes, it's a type, it's a modification, but it's a process as well. So there's a distinction. I can modify and not have anything to do with gene drive, okay? But if I want the trait to spread relatively quickly, then I modify and I incorporate a gene drive, all right? So two things. All right. So the number 2 is where the gene drive comes in, okay? Now we are all together. It's about figuring how to spread things quickly through nature, all right? So if we take a step back, I said these are some of the ways we could modify so that, you know, we are reducing the capability to transmit the disease. We can create resistance to the parasite in the mosquitoes. We could modify to maybe increase insecticide susceptibilities in case the population were resistant. We can do that. We can try to do behavior change, and we can also, you know, do other things. But all these things that if we did them, that's fine. The problem we'll face is that if we now tried to have those spread and we just bring a modified one like that and try to spread that trait in the wild mosquito populations, very quickly we will encounter a problem, because first, your numbers are few compared to what's out there. So even if you started it, at most, because of the way the laws of inheritance work, if you remember the Mendelian approach, is that that trait of yours is likely to be spread to just half the progeny if it came in as a heterozygote. So therefore, with that trait, the other ones are more than yours, so they'll keep spreading their traits, which you don't want, and down the road, at least what will happen is that your frequencies of your modified mosquito, either they remain the same as you introduce them, or they can even decline, because if it happened that your trait that you introduced is not that beneficial to the mosquitoes, and there's no reason why, for example, creating anti-parasite resistance or even susceptibility to insecticide can be beneficial to a mosquito, that actually harms it. So natural selection tends to weed out those kind of things. So your modification will be facing what they call selection pressure, and you're likely to lose it. So if you want to go around that, you need to add a drive that will help you overcome that, okay? So we are there. This is just the Mendelian approach, whereby for things that are diploid, which have 2 copies of a chromosome, one from one of the parents, to spread that trait, yeah, to the progeny through the gametes, of course, there's a fair chance, a 50-50, that one or the other is passed on. So it's 50-50, yeah? And at that rate, the frequency really doesn't change. It remains the same. The only way to effectively use this approach is if you were to create many, many more than the wild mosquitoes, and then you overwhelm the numbers. But that's not very practical for a disease like malaria. Yeah, you can do that for a disease that's limited, maybe that's geographically limited, maybe in the cities. But if you think about malaria, rural, the distances and the numbers involved, too much. You can't do that. Okay. So then gene drive, that's how it comes in. So we said under Mendelian, a trait, mosquito with a modified trait that's heterozygous will pass it on just half the progeny, okay? And so with this kind of inheritance, you can clearly see down the generations, your trait is, the frequency is not increasing, okay? And you could even lose it. So what you want is this here, the gene drive, whereby if I introduce a trait, yeah, then it's preferentially transmitted to the offspring. So with a drive, then you have a scenario whereby a modified mates with a wild type, and most of the progeny are carrying that trait. Yeah. So if this is what you have, then theoretically you can spread your trait relatively quickly through the wild mosquito population, and then that's how you can make genetic modification a viable tool for malaria control. Without a drive, you'll be forced to have numbers that have hopefully many, many more than the wild mosquito population, which is a hard task. Then you're saying you need lots of factories, you need to keep releasing more than a single generation, etc. But here, this is more self-sustaining, and that's why it's a bit novel and slightly different, and makes the vector sort of help you in the control itself, because it's spreading the trait for you. So that's where the innovation is, and that's where the gene drive comes in, to spread the modification. So again, this is another cartoon. It's to increase the spread of the modified gene that you had. And now, of course, the gene usually is anchored to a trait that we want to spread. And if that trait was, let's say, creating female infertility, then that's what will spread through the mosquito population. And if you have many of them being infertile, then they don't produce many offspring, so then the malaria transmission can be curbed. Or if it's anti-parasite resistance, again, same thing. The drive spreads that kind of trait quickly through the mosquito populations. So then the other thing is then, where does then drive come from? Yeah. So luckily in nature, there are already examples that show drive, yeah, that the scientists can use as a basis for making their synthetic versions, yeah. So we know selfish genes, and there are a number that have been described by molecular biologists. So you have, for example, transposable elements. They like a way of inserting themselves in different locations of the genome where they were not, so they're mobile. You have what they call chromosome distortion mechanisms. These have been described in many insects. What they do is they try to destroy the competing, remember they're diploid, so if there's a counterpart of the chromosome that doesn't carry that, then during meiosis somehow the one that has the trait destroys the other one, so that in the transmission it's only the one that carries the trait that's driving that goes through. Sort of selfish. So it's like genes are also fighting among themselves. And then you have nucleases. The most common ones that are used here would be those which spread by a homing. So they home to a location, and then they copy themselves in that location where they were not, using a template of where they came from to do the repair. So there are those, and that's what really scientists do, and that's what they harness, and these things are already, in nature. If you look in the microbes, the bacteria and the viruses, so you find, for example, these nucleases, yeah, and some of them can be selfish. So scientists just use those, and then they harness them, and then they make them to work for them in the mosquito. So in a way, it's synthetic, but it's still, based on nature, yeah. So, yeah, so here this just wraps up how to envisage gene drive if you compare with other methods that are out there that could have something to do with still modification, but maybe without driving. So the most common technique that you'll hear is the SIT, sterile insect techniques, and in this you can still, of course, introduce modification either by irradiation or a number of ways, but you can also genetically modify and create that sterility. But that doesn't make this gene drive unless if you put a drive component to that. So if you just left it at this, it's a classic SIT. It's genetic modification, but remember it's not spreading relatively quickly through the world mosquito population. So what that means is you'll still have to keep releasing many generations, and you have to have enough numbers to really compete with your wild type. Okay, so that's what it is. And then the others who have looked at Wolbachia, this is not in malaria vectors, but some other vectors, and it has worked for them. But again, this one also creates some bit of incompatibility, and that trait can spread itself. And that's what the dengue people are mostly relying on in Asia and the Americas. But that's not for mosquito control. We've tried, we haven't got really Wolbachia that we could use. So for now, that's not available to us, but people are still looking. But this is different, this is not really gene drive. It could be a Wolbachia drive, but not gene drive, it's a different drive. But the gene drive itself, the characteristic that you want to take away from here for it is that you modify and you find a way of incorporating drive in the design of that modification so that it's preferentially inherited by the offspring. And if you do that, it makes that now self-sustaining and a practical tool for use of the malaria control, because you can take your modified mosquito, they don't have to be many, you can release them even in a remote area, and then they'll spread relatively quickly, yeah? And hopefully over time, that trait will become frequent enough to start making an impact in the malaria control, okay? And we think this is the best way to control diseases that are in remote areas where you have logistic challenges, where you may not have lots of capability to produce some of these things. So there's a niche that gene drive can help, but it doesn't mean that gene drive alone is the one that's going to solve everything. So it still needs to be employed in complement with the other existing measures. So the features that we really see that gene drive brings on board is it could be long term. Of course, it can be safe because we say genetic modification quite specific. Yeah, and then of course the resistance that you encounter using other methods will not be the same kind of resistance. So you can still bypass that and use this. Of course, it can be applied in areas where maybe the health systems are strong or they're weak, because it doesn't depend so much. Once you seed it, the idea is it can keep spreading and doing the malaria control for you. So if you have areas, for example, like when we had COVID, and then For example, the malaria control program had to take a backseat to the COVID control. Had we had a gene drive, it would have continued working, suppressing the vectors, and maybe we would not have come out of COVID the way we came out of Warsaw for malaria and other things. So there are those kind of things. And of course, we haven't done the actual costing now, but we think it can be cost effective, even at low prevalence, and you don't need people complying or adhering to your control methods. If you give out nets, people have to sleep in them, and then you have to fight with them, tell them, go under the nets, sleep under the nets, don't misuse the nets. But this one, the mosquitoes just do it by themselves, so you don't have to worry about that. This one also doesn't depend on the economic status of the people. Some of the other methods, if you're rich, then you can afford them. If you're not rich, then you're on your own. But this one is more egalitarian. So the advantages this can bring to the malaria fight. Yeah, but I can't just stop there. Of course, I've painted a very good picture, but it's also good to say, of course, there's still issues, even with gene drive, like all other things. So, of course, the mosquito is going to fight back. Just because you can just quickly relatively spread the trait, it's still a genetic approach, and it still targets somewhere, and the mosquito can also counter that by, of course, creating, variants there so that the gene drive can't quite target specifically as it could, and then that could stop it. So that's also possible, and you have to be wary of that. But then there's also the issue of governance, regulation, and public acceptance. So for here, we are here because of the regulation. So gene drive hasn't been done anywhere. So then how do you regulate it and things? So we have to build that and make sure we have robust systems for their regulation, also the governance, and then there's the acceptance for them in our populations. And then there's always the risk as analysis, that kind of thing. We talked about this a little bit yesterday, about a potential effect on the biodiversity and unintended ecological consequences and things like that. So again, as we design those things, we have to have those things in mind. So all those things come with gene drive, but just because these things are there doesn't mean we cannot be bold, and we cannot contemplate, and we cannot design them as a tool. We can design them, and we can look at each of those things case by case and make a decision. It's the same thing that happens when you have other tools. They're made, be it a vaccine, be it a drug. It goes through a trial. We find, are there side effects? Can they be managed? If they can and they cure, then you don't just drop it because of side effects. You go on, you find a way of managing those things. So it's going to be no different with gene drive. So in summary, let's just understand that gene drive itself is a natural process, because it happens in nature, it's around us in the microbes, but we can harness it for vector control. And that's how humans work. You see something in nature, you copy it, you use it for yourself. But we also know that gene drive is not this super weapon that it's made to look. It can be countered, you can have resistance to it too, and even as you design it, you have to be wary of that. And at the moment, we don't have a gene drive product that's ready now for use. All these things are still in the lab research stage. So we're talking about that. So there's not tool out there. So if you hear releases happening, like you hear in the literature, people say, the news mostly, people say gene drives were released in South America, Asia. No, those were not gene drive. Those were Wolbachia things, or they were Oxitec, modified but not driving. So we make a distinction. There's gene drive, and then not all modifications are gene drive, and they don't need to. And then the way the gene drive development is running, most teams are using a step-by-step approach, because we know there's still a lot of things that need to be put in place. So it's a staged approach to development. So they don't create a gene drive just right now, suddenly. They go through steps, first versions that are not driving, but having the characteristics that you want. You first test those, convince yourself, that the assumptions are right, and it's working the way it's been intended to work before you start handling the one that will be driving. But there's also infrastructure upgrades that you have to do, and lots of other things. So we're still a long way to go. So I think that brings me to an end of gene drive, but let's just say gene drive is a process, and you can have modification that don't have drive. And that's okay, it really depends on what you want. But for vector control, for these vectors like malaria, which are widespread, which are numerous in remote areas, gene drive will give you an advantage if you incorporated it into the design. So I'll let Mamadou and Fred chip in case I've distorted anything or I forgot anything, and even Brian, because they too are gene drive people.